Let's now talk about the synthesis and metabolism of acetylcholine and catecholamines. Acetylcholine is made from one set of neurons referred to as cholinergic neurons. Cholinergic neurons release acetylcholine. How do they synthesize acetylcholine is our focus here, so let's talk about it. You need two substrates, choline and acetyl-CoA, and the two are brought together within the vesicle that is found within the synaptic knob of a presynaptic cell, and then the combination of these two substrates results in the formation of acetylcholine. Whenever there is an action potential and the release of calcium occurs at the axon terminal, then the vesicle is moved from its original location to the cell membrane location. So here, the membranes of the vesicle as well as the cell membrane come together and create an opening, and that is how acetylcholine is released. Soon after acetylcholine is released into the synaptic cleft, the clearing of acetylcholine happens immediately. This is to avoid unwanted stimulation of the postsynaptic cell by acetylcholine. So the clearing of acetylcholine is by the release of an enzyme, acetylcholine esterase. The enzyme basically splits acetylcholine into choline and acetyl-CoA, which are recycled to synthesize new acetylcholine. All right, we could take a look at pathological situations where you do not have sufficient amount of acetylcholine esterase released. In this case, you would have acetylcholine remaining in the synaptic cleft and activating the muscle fiber more than normal, and then that leads to spastic paralysis. Then other immune disorders are where you may have acetylcholine receptor blockade, and therefore you have acetylcholine, you are not able to bring about its effect at the postsynaptic cell. That leads to another type of paralysis, that is flaccid paralysis. There is no contraction that is triggered by acetylcholine because of the absence of its receptors. So then there are drugs used to target acetylcholine esterase function so that you can manipulate the presence or absence of acetylcholine, and then therefore you can control muscle action appropriately. Muscle relaxants that are used during surgery, for example, may require the use of depolarizing and non-depolarizing type of muscle relaxants. They could target the action of acetylcholine esterase. All right, so good. With this, let's move on to talk about catecholamines. What are catecholamines? A group of neurotransmitters that involve dopamine, norepinephrine, and epinephrine. All three are made from one amino acid type, that is tyrosine. All right, tyrosine is first converted to dopa, that's like a precursor, and then that would become dopamine, and then dopamine is converted to norepinephrine and ultimately epinephrine within the vesicle. And they are all catechol rings with small changes in the structures. For example, a methyl group added to norepinephrine becomes epinephrine, and so on. All right. Now, the neurotransmitters here are metabolized by using enzymes such as catechol-O-methyltransferase, COMT. This is the same one for dopamine breakdown as well as norepinephrine breakdown. Monoamine oxidase, MAO, is another enzyme that is utilized in the breakdown of these neurotransmitters. There are subtypes of MAO, MAOA specifically for norepinephrine, and then MAOB for dopamine metabolism. All right, norepinephrine is important, are associated with depression. So in patients with mental depression, you may use drugs such as monoamine oxidase inhibitors, MAO inhibitors, so that you have norepinephrine in larger amounts for a required amount of time. Okay, so all these agents, enzymes are metabolites. For example, DOPA is the metabolite that could be given instead of using dopamine, because the DOPA crosses the blood-brain barrier, but not dopamine. That's where, the choice of drug is where you may want to think about their intermediate metabolites than the neurotransmitter itself. All right. So in any case, Parkinson's disease is where you do need the replacement of dopamine. That is where you may see the use of DOPA instead of dopamine directly because of the fact that they cross blood-brain barrier or not. Okay, so anyway, this is how these neurotransmitters that are associated with the autonomic function are made, are metabolized. All right, we will stop at this. Thank you very much.